Change the name of your solution. We change the name of the ligand solution to the lot number of the monoclonal anti-chimeric Claudin-18.2 antibody, and then we change the name of the regeneration 1 solution to 10 millimolar glycine HCl pH 1.5. Now click on Send to Queue. We will now plate our solution. You can follow your own plate map, but in this video, we will add 60 microliters of 3 microgram per milliliter monoclonal anti-chimeric Claudin-18.2 antibody from earlier into a 96 well plate in well C1. Add 60 microliters of the regeneration buffer to well C2. Add 60 microliters of running buffer to wells H1 and H2. You can now place the plate on the plate rack and the rack on the rack tray. Place the rack tray in the machine. On the bottom right screen of Send to Queue, click Open then Ready to Start. Select the folder that you want to save the data in and rename the file. Once you hit Save, the instrument will start to run. After the program is running, click Runs above the main interface of the BioCore AK control software. Find the result file, double-click or click the Open button in the lower right corner to open it. Select Sensorgram. The entire coupling process can be viewed. Add 212 microliters of running buffer into well A1 of the 96 well deep well plate. Add 49 microliters of Human Claudin-18.2 His twin strep tag into well A1 and mix gently. We are going to perform a two-fold dilution. Add 130 microliters of running buffer to wells A2 through A8. Add 130 microliters of A1 to well A2. Pipette mix. Add 130 microliters of the solution in A2 to well A3. Pipette mix. Do this for the rest of the wells until well A8. Remember to change the pipette tips in between each dilution. Pour the running buffer into a basin. You can follow your own plate map, but we will be using a multi-channel pipette to add 110 microliters of the running buffer into wells C1 through C3 of a new 96 well plate. We will call this Plate 1. Using a multi-channel, pipette 110 microliters of the two-fold dilution from earlier into the 96 well deep well plate into wells C4 through C11 of Plate 1. Add 240 microliters of the 3 microgram per milliliter monoclonal anti-chimeric Claudin-18.2 antibody solution into wells C1 through C2 of the new 96 well plate. We will call this Plate 2. Add the regeneration buffer to wells C3 through C5.